Good afternoon, everybody. I'd like to just introduce myself. I'm David Woodland, and I'm the Chief Scientific Officer for Keystone Symposia on Molecular and Cellular Biology. And I really want to welcome you to this panel discussion on HIV/AIDS strategies for an endgame. And I have to say I'm extremely grateful to Dieffenbach for agreeing to moderate this event. And I really want to thank all of the panelists who are here today. And with that, I will hand over to Carl to get the show rolling and to introduce the panelists. Carl.
Thanks so much, Woody. And I really appreciate seeing this audience as full as it is. What we're looking forward to today is an active dialogue, not just among ourselves, but with all of you. That said, could I have my slide? To build off what Woody just described, the theme of the video event, the strategies for an endgame, I think it's really important that as scientists we can focus the discussion today not just around the strategies toward the endgame, but consider the strategies in the context of the research continuum. Given the situations we face today, it is essential that we be able to balance the research across the continuum. So what is the appropriate balance? And is there such a thing as an appropriate balance? Everything from basic science to efficacy to proof of activity. We felt in earlier discussions that for today's activities, we should really try to focus, because this is where all of us tend to live who are in this room, in the first three boxes, the balance between basic science, the translational work, for proof of concept leading to evaluation for efficacy. So we can focus on those three. I imagine we will have some spillover into real life implementation, but I think that will be the minimum activity. So in terms of the types of activities that we could consider within the research continuum are the obvious things that we've been talking about at this meeting. How do you make a safe, effective, and durable HIV vaccine? How do you prevent HIV infection? How do you cure HIV infection? And what are the large remaining questions in HIV pathogenesis that need to truly be tackled? So what we've asked each of the panelists to do is to stake a position. And in some cases, we've asked people to really go out on a limb and take relatively extreme positions with the goal of inciting discussion and debate. So at the end of this, if you feel somebody has been a little bit out of line or has taken an extreme position, please keep in mind that they were probably doing that for the purposes of keeping this discussion lively and interesting. So everybody will be able to disavow whatever they say based on that, and so I've tried to give everybody the appropriate fig leaf. So what I'd like to do is first call on Galit, who will start us off and then provide sort of a perspective for 3 to 5 minutes, and then we will march down the panel. And when we finished with Peter's 3 to 5 minutes, we will each have thought of questions, and we will ask them to each other. But in the meantime, I actually want all of you also to be thinking about questions, because ideally, I want you all to participate, because this can become the strength of having it live like this, and why it would be the thing that we can do that's different than the video broadcast is there was no live audience. You're the live audience. You're not just here to be passive, but to also be active participants. So with that, I'm going to turn it over to Galit and take it away.
Thank you. So I'll take 2 positions. I'll wear 2 hats here. First, as one of the conference co-organizers, I think that Nicole and Susan and I, as well as Victor and his co-organizers, had a good time this round of planning, specifically because we could bring people together from different fields, and particularly in the vaccine field, bringing the basic immunologists together with those who are doing the in-human vaccine trials, phase 1, phase 2, and beyond, and to really create atmosphere and opportunity to potentially reprioritize the way we think about advancing science. And that's really what we hope to do also with the vaccine and the cure group. Now, my extreme position comes as a young investigator, and here's where I'm going to try to set the first potentially controversial ideas out there. So we all know that humans make the best models for humans. We know that we need to test concepts in humans, understand human immunology, and there is a need to move concepts forward so we can understand how the human immune system will respond to an immunogen or therapeutic in either cure or in vaccine. But what I would argue is that the large pots of money that we put into doing phase 1s and phase 2s with concepts that we're just recycling over and over again with a very narrow range of new concepts moving forward is sabotaging our ability to do discovery and to really start to funnel and fuel all the innovative, new, transformative products that can move forward to really make a huge impact in moving the field forward. So what I would say, and I hope that my co-organizers will, or co-panelists will disagree with me, what I would say is that we are preventing young investigators from making it, from bringing in new people from the outside because we're putting money in to make incremental discoveries. And just to look at, how does a T cell do something, or how does a B cell do something, instead of really trying to push ourselves and really push the whole field forward and start making those cutting edge discoveries that may cost a little money as well, but could push us much faster.
Wow. Yeah. That's a perfect start. So, David, no pressure.
No pressure. Well, so we've started off in a very different direction than I thought we were going. And so I feel obligated to respond in some way. And I guess in response, I would say that I don't totally disagree. I think that these are good points. I think at the same time, there are also advantages of the current system, and that I think we've seen some exploratory sort of wild idea pots of money funded in various areas in the recent years that have really not, at least as far as I can tell, brought very much forward. So I think that it's important to focus on novelty and discovery and to figure out how we can fight against the sort of forces that bring us back to sort of safe middle ground. That investigator, they can do aim 1. That's good enough. I think really the major problem in the field is the commitment of governments to fund science. I think that's the root cause of all evil. And I think that perhaps we should be doing a better job of advocating on the political, social side for what the importance, what is important that we do from our work that brings benefit to everyday life and to human society. So I won't totally disagree, but I won't totally agree either. So, your turn.
Thank you. Thank you. So let me just make a couple of points. One of them about human clinical trials, and I think no one will argue, I hope no one will argue with me, that I think testing an immunogen in a clinical trial really offers the ultimate proof of activity. It's expensive and costly on many levels, but I think we don't actually get the ultimate answers until we test it in humans. But I do agree that we need to balance that by being nimble and responsive in how we react to the advances in the basic science field. And oftentimes that doesn't happen quickly enough. So we really need to sort of shorten that axis of the time from proof of concept to proof of activity, and sort of thinking about nimble manufacturing processes and how we produce immunogens that can be tested quickly in parallel, phase 1 studies where we can even sort of do some immunogenicity evaluations in real time so that we really sort of get very quick readouts about products and sort of a go, no-go decisions about advancing to more advanced clinical phases of testing. And then just, another comment is about sort of as we move forward and think about designing even more complex combination prevention studies, we really need to go back to and think carefully about the animal models and basic science and make sure that we test hypotheses that would sort of put forward the most compelling combination prevention combinations that can be tested in human clinical trials, and make sure that we test sort of novel approaches and novel platforms and delivery mechanisms, and that also will help the implementation science. And I know that we're not focusing on that right now, but I think just to keep that in back of our minds.
Well, thanks. I think, in the end of the day, this discussion is there's only three things we can do. We can treat HIV better, we can cure HIV, or we can prevent HIV. Those are our three options. And Galit kind of lays out novelty, which is great if novelty is sufficiently compelling that we can act on it. And then the alternative, the far alternative to novelty, is just like make work. Like just keep people going because that's their life's work, and even though it's no longer interesting, they should just keep going. And that's a tension that as an older guy just wants to keep going. It seems good to me. But I know. But the best thing. But there is focus and tenacity. Okay. So in other words, these things take a long time. For the kind of home runs don't occur your first time at bat. Most of the big things were incremental and were built on a platform of. Yeah, sure. No, not you can't argue right now. But a lot of things were a lot of things were not incremental. They were tenacity with sufficient biological plausibility to be compelling, and then $100 million invested in proving that the thing worked. So that's part A. And part B is, at this point in time, again, treatment, prevention, and cure, it's like we have an obligation to try and advance this as fast as we can advance it while not skipping in the middle. To me, if you look at 85, we didn't know what the cause of this disease was. 95, there was one drug. 2005, there were 3 drugs or 14 pills or whatever. And 2006, it was 1 pill a day. I would be shocked if the field did not have a major change in the next 5 to 10 years in terms of treatment and cure. That built on kind of everything that's happened to date. I would be shocked, and Galit, you can argue this. I'd be shocked if we don't already have some fundamental information that will change treatment for infected people, that will simplify their treatment. And I hope that happens. If somebody hits a home run, that's fantastic. But I would argue for tenacity and focus. I'll stop.
Tom. So I take the view that I want it all. And that is research and practice go hand in hand. It's constantly in evolution. And it's a dynamic process. And maybe it's tenacity at that dynamic process, but it's a full continuum as shown on that graphic. And let me just give you a couple examples. So in the treatment arena, you started out with AZT. It added 6 months of life. And you could have stopped there and just said, how do I scale this up to more people around the world, AZT? But instead, the research arena kept going right from the basic science, right up into protease inhibitors, integrase inhibitors, receptor blockage, and it continues to this day. More and more, we're going to have better and better treatment. We can now treat people with 1 pill once a day around the world for $300 a year. Hepatitis C will have to follow that kind of model, and it's not there yet, but it will. And the only way you do that is that you don't stop. You're not satisfied. You need to keep pushing the envelope as to how do you get better treatment to those people. And that then leads into cure, because you're going to treat all these people for the rest of their lives, more and more people getting infected. 2 million people got infected last year. And so it's a never-ending story unless we build to the cure. And one of the best examples for that would be prevention of mother-to-child transmission. And where there are hints, almost proof of concept, we might be getting cure, whether it's functional or otherwise, in those children. And so we need to better understand the reservoir in the children, how best to affect that reservoir, and continue to push that envelope. On the prevention side, obviously the biggest disappointment has been vaccine. But even though it's been a disappointment, there have been hints of successes, such as the Thai trial, and I think we should build upon some of those and go back to proof of concept, go back to basic science. The session this morning on vaccine with the different types of modalities was just absolutely fascinating. And I think we need to encourage all of that to come forward so we can build towards that better and more effective vaccine. Some of you know I did work in the circumcision field. Well, there were observational studies that showed that it was protective. We did the randomized trials, took it through the efficacy trials. Now we're trying to scale it up. The problem is towards the end. How do you really get that to be adopted to individuals who could be afforded that type of protection? And as I walked around the posters today, I could see that individuals were still trying to understand why would the foreskin afford some protection once it's removed. And yet it's through that basic science that we learn about the interface between the macrophage dendritic cells, the activated CD4 cells that are within that tissue, and how we might be able to draw analogies then from the animal models to humans. So Carl, I'd like to keep the whole continuum going forward. I don't see any reason why we have to cut back on the basic science just because we have some efficacious trials that we're trying to scale up. I think we have to continually prove to do better at all ends of the spectrum. Peter.
Well, you set me up perfectly. Again, remember, we're asked to take polarized positions, but that's all very nice when there's no limit to the budget. The truth is, we've got a cap. And when I started, a 20% funding rate, 19% funding rate was fine, but at 6 to 7 to 8%, we've got to have, if you're balancing your home budget, you got to figure, can I afford this vacation or not? Do I have to cut back? We're at a point now where I think part of this actually has to come into practical discussion. We would all like to have everything, me especially.
Yeah.
But in this limit where we're at now, I think some important questions need to be asked. And as an example, our group was involved in the first cell genesis gene therapy trials looking at mucosal quantification as a reservoir. We designed the Sangamo mucosal compartment. 10 years ago, we published a paper showing the reservoir stays there no matter what at low levels. That doesn't get much attention, that's fine, but it's there. Truth is, I have to ask, why are we spending all this money on a cure? I know, I was paid to say that sentence because it needs to be just put up front, but there's a whole lot of money going toward a cure that might be being able to be directed back toward some of the questions I happened to put up yesterday was, we don't even know what cells are getting infected. We don't know what the responses are on the innate sides yet. And so some of these will help us to divine new technology or new approaches, new interventions that we are missing in some of the vaccine trials that go all the way to phase 3, and then we realize they don't quite really work. Microbicide trials as well. Maybe some of these earlier structures, and this falls under what was suggested by Tony and Carl in 2008, very intensified trials. And I would advocate here is when we do these early exploratory or phase 1 trials, by definition, the subjects enrolled have to be extremely well characterized. Well, if we always take extra samples, then these are put in a bank that are very well characterized for people to use these samples to ask additional questions that weren't even part of the initial protocol, but they are there to begin to do some of these hypothesis-generating surveys that then allow you to go to hypothesis selection because you actually have some basis, which then allows you to propose the hypothesis testing R01. In a limited budget, I would advocate this goes, and it also goes back to the way we started of keeping young investigators having a slightly promising future that maybe there's going to be funding. All right, how about full circle? You set me up. Thank you.
Yeah.
Thank you for that. I think that there were a couple of themes that emerged from that, and I'm going to challenge my friends on the panel that by and large there's a fair amount of wishful thinking going on. Is that at the end of the day, and this is something I live with all day, every day, and have for the last almost 10 years now, we have a flat budget. And that means that with every year, there's less buying power. That's the reality we're in. So we have to actually make very difficult decisions about the balance between basic translational, and then the evaluation of efficacy. And I guess that's one of the things I'd really like us to get down to with this is, are we too heavily weighted toward translation and toward efficacy? And do we really have the basic knowledge that we need in vaccines, in microbicides, in cure to really move forward much beyond, say, some very small proof of concept trials where we're doing those kinds of first-in-human experiments where we can really get at basic mechanisms? I think that is really the type of question we need to be asking and answering. And the other issue, if we take by analogy what happened with the drugs, I mean, Mike made the point about we've gone from, and so did Tom, we've gone from AZT to multiple fixed-dose regimens. The reason was industry saw an opportunity and jumped in and took it away from government to develop these drugs. And so if we think about an alternative model, what is it that we need to do as scientists to generate that proof of concept that makes industry say, there's something here that we can not only do good for the people of the world, but do good for our shareholders?